Good day, everyone. We are Farmaceuticals, and here's our final presentation on our journey towards choosing the best cloning technique for our gene of interest, which is waxy gene A or WXA. We will be first giving a brief background on the waxy gene A. The WXA allele of the waxy gene produces higher granule-bound starch synthase 1 or GBSSI enzyme activity, which increases the synthesis of amylose in the rice endosperm, usually around 20 to 30%. Because of this higher amylose content, rice varieties carrying the WXA allele have a firmer, less sticky texture after cooking. Now let's dive into the functional analysis of waxy gene A. So the figure on the left shows the starch synthesis in the endosperm wherein the production of GBSSI can either increase or decrease amylose content, thus changing the quality and texture of rice. Moreover, the function of WXA is affected by the insertion of transposable elements that results in a change in the gene product. Meanwhile, the figure on the right shows how AGPase makes ADP glucose, which is then used by the GBSSI to elongate amylose chains. When the fully functioning WXA gene allows efficient splicing, it produces high GBSSI expression and higher amylose synthesis. Now for the gene expression. So, as mentioned earlier, WXA is highly associated with the endosperm in seeds where the enzyme GBSSI is synthesized. Moreover, gene expression is higher during the early to middle stage and decreases as the seed reaches maturity. Additionally, high temperature during grain filling results in chalkiness, which is a product of irregular starch deposition. Moderate salinity stress also downregulated the WXA gene, resulting in a lower amylose content and contributed to the increase of chalkiness in the rice grains. Now proceeding to the gene sequence, presented on the screen is the genomic DNA of WXA from RAPDB, wherein the untranslated regions are highlighted in gray, splicing sites in pink, and exons in blue. This sequence was used for the future cloning processes. Presented on the screen are the restriction sites of WXA as identified by NebCutter. Restriction sites are the DNA sequences that can be recognized by restriction enzymes. In comparison with the common cloning vector PUC19, the highlighted restriction sites were found to be common between the plasmid and in WXA gene as identified by NebCutter in the previous slide. Thus, the unhighlighted ones can be used as restriction enzymes for performing cloning techniques.
Now let's talk about the different cloning techniques that we did in class. So first up is the restriction cloning, which utilizes restriction enzymes to cut the vector or backbone plasmid and insert our target DNA sequences. So it starts with identifying restriction sites of our restriction enzymes, and then we design primers for our target DNA sequences according to these restriction enzymes, and then we amplify it using PCR. And then the PCR products and the vector plasmid will be ligated together, and then the resulting clone plasmid will undergo AGE to verify its content. Displayed here are the different clone plasmids for our promoters, CDS and gDNA. And the fourth picture shows the gel from our AGE, where, where these plasmids are digested using their respective restriction enzymes. They're showed two different bands, one for the PUC19 vector and the other for the insert.
3A cloning is an efficient cloning method that bypasses the need for restriction enzymes by taking advantage of Taq polymerase, which adds a single A residue to the 3' ends of PCR products. These A-tailed fragments can then be ligated into vectors containing complementary T overhangs. In this case, the WAXA promoter, CDS, and gDNA sequences were amplified by PCR and inserted into the PCR 2.1 topo T vector. Successful ligation was confirmed by agarose gel electrophoresis, with both the vector maps and the AGE results showing that all three sequences were successfully cloned, as indicated by the products observed in lanes 5, 6, and 7. Similar to TA cloning, topo cloning is a restriction enzyme free method that uses topoisomerase 1 to both cut and ligate DNA. In this project, a directional topo approach was used in which the insert carries a 5' CACC overhang and the vector carries a complementary 5' GTGG overhang, ensuring that the GOI is inserted in the correct orientation. Primers for the WAXA promoter, CDS, and genomic DNA were designed with the required CACC sequence, and the resulting PCR products were cloned into the PBAD202D topo vector. Successful cloning was verified through AGE, followed by additional confirmation using colony selection, antibiotic resistance screening, and blue white screening. Both the vector maps and the AGE gel indicate successful cloning for all three constructs, as shown by the distinct bands for each GOI and the PBAD202D topo vector in lanes 5, 6, and 7. Antibiotic resistance screening ensured that only cells containing the PBAD plasmid, which contains the ampicillin resistance gene, survived. Non transformed cells were removed. This was followed by blue white screening, where successful inserts disrupted the LAXA gene and produced white colonies, while empty vectors produced blue ones. The white colonies were selected as the positive clones.
Gateway cloning is a site-specific recombination system that transfers the DNA fragments between the plasmid vectors using ATT sites, and it uses a BP and LR reaction. In this experiment, we used the promoter, gDNA, and CDS sequences. They were amplified using the ATTP primer with the ATTP sites of the donor vector using the BP reaction to create the entry clones. And these entry clones were recombined with the destination vector through the LR reaction to generate the expression clones. To verify the products, the restriction digestion simulation was used to identify if the enzyme cuts the vectors and the inserts, which also follow their fragment size. And the colony PCR of ampicillin resistant plasmids was done using the M13 forward and reverse and the T7 forward and reverse primers to confirm if the insert's presence is in both the entry clones and the expression clones, and it is seen in these results.
Gibson assembly joins multiple DNA fragments in a single isothermal reaction, relying on three enzymes working together: the exonuclease, DNA polymerase, and DNA ligase. In this study, the vector PDES was linearized using the restriction enzyme XBAI. Inserts including the CAMV35S promoter, WAXA CDS, and the mChari reporter gene were prepared through PCR. Primers were designed with 15 to 20 base pair overlaps and appropriate GC content. The Gibson reaction proceeded at 50 degrees Celsius. The resulting plasmid was transformed to CCDB survival E. coli and grown on ampicillin and chloramphenicol containing plates. Results showed that two expected bands were present: a 6184 base pair band for the PDES vector and a 3192 band for the insert cassette, confirming successful ligation. Colony growth on selective media and the red fluorescence from the mChari further verified that the construct was correctly assembled and expressed. Infusion cloning relies on the infusion mix, creating 3' overhangs on the linearized DNA. After assembly, the construct is repair naturally by the DNA machinery of the E. coli after transformation. PDES vector was linearized using XBAI. A cassette insert contained the promoter, the CDS, and the reporter G. Primers are with 15 base pair overlaps and appropriate GC content. The reaction occurred for 15 minutes before transforming into CCDB, survival E. coli, and plating on selective media. Restriction digestion produced two bands at 6,192 base pairs and 3,192 base pairs for the vector and insert respectively, suggesting the successful assembly. Colony growth on the selective plates and the visible red fluorescence from mChari further confirmed the insert was incorporated and expressed.
The study identified candidate sgRNAs by scanning the target sequence for PAMs recognized by the SP-Cas9. Here we evaluated 5 designed sgRNA candidates for CRISPR-Cas9 targeting based on 4 key parameters: on-target activity, off-target risk, GC content, and homopolymer runs. Now, on-target scores range from 51 to 77.2, reflecting predicted cleavage efficiency. Off-target scores also varied widely, which emphasized the potential for unintended edits, with the lowest risk observed in the fifth sgRNA. GC content was also assessed to ensure stable and specific binding, ideally between 40 to 60%, while all candidates were free from long homopolymer runs, minimizing sequencing errors. Considering all criteria, the second sgRNA was the most balanced sgRNA for specific and efficient gene editing. After selecting the optimal sgRNA, we simulated the CRISPR-Cas9 cleavage mechanism in silico. The sgRNA Cas9 complex binds to its complementary genomic target adjacent to the PAM sequence, and Cas9 here introduces a precise double-strand break, 3 base pairs upstream. And now, in the absence of a donor template, repair occurs primarily through the error-prone, non-homologous end joining pathway, which often generates insertions or deletions at the cut sites. If such indels occur within an exon, they can also cause a frame shift mutation, resulting in premature stop codons and truncated non-functional proteins, achieving a gene knockout. After selecting the optimal sgRNA, we simulated the CRISPR-Cas9 cleavage mechanism in silico. The complex binds to its complementary genomic target adjacent to the PAM sequence, and Cas9 introduces a precise double-strand break, 3 base pairs upstream. Now, in the absence of a donor template, repair occurs through the error-prone non-homologous end joining pathway, which can generate insertions or deletions at the cut site. Now, if these indels occur within an exon, they can cause a frameshift mutation, which could result in premature stop codons and truncated or non-functional proteins that could achieve a gene knockout. Now, to facilitate experimental validation of CRISPR-Cas9 mediated knockouts, primers were also designed to amplify the genomic regions flanking the predicted cleavage sites for each sgRNA candidate, targeting the WAXA gene. For instance, the chosen sgRNA was paired with the left primer and right primer, positioned several nucleotides upstream and downstream of the target site. Similarly, primers were designed for the other sgRNAs to ensure that any insertions or deletions generated by the non-homologous end joining repair pathways could be captured during PCR amplification.
Among all the mentioned cloning strategies, we came down to choosing between the strategies that have a high accuracy and modern approaches, which are the Gibson assembly and the infusion cloning. Gibson assembly is more suitable for assembling large fragments because even if there is a risk of errors, it ensures the ligation between fragments because of their long nucleotides. And infusion cloning is the best choice because of its practicality for this gene, since the essential fragments for the waxy gene A is a CDS portion. And with this specific fragment, we would avoid errors in insertion, and it does not also require the use of DNA polymerase and ligase, making it less prone to errors and scars. And again, plants engineered with this waxy gene A cloning method would have enhanced grain quality, especially for crops that naturally lack the optimal conditions to produce it efficiently.